This is The Future Of, where experts share their vision of the future and how their work is helping shape it for the better.
I'm David Carston. Insects are vital to healthy natural and agricultural systems. They pollinate many of the plants that humans and animals eat. Services that ensure adequate plant pollination add more than $2.6 billion to Australia's economy. But insect pollinators are under threat by rapid climate change, habitat loss, pesticides, and emerging pests and pathogens. With me today to explore this topic is Associate Professor Paul Neville and Dr. Joshua Castell. Paul leads the eDNA MineSite Biomonitoring Research Group at Curtin University. Joshua is a Curtin PhD graduate and researcher at Cambridge University, where he is examining insect pollinators using eDNA. Joshua and Paul have found that eDNA, or environmental DNA, is a powerful tool that can accurately detect insect species interactions with flowers. This technology can play a crucial role in sustaining important insect-mediated ecosystem services like pollination and, in turn, the food and nature systems on which we rely. If you'd like to find out more about this research, you can visit the links provided in the show notes. Paul, apparently 40% of all insect species could be at risk of extinction. How did we get here?
Yeah, it's a really good question, David. There's four main causes for the loss of insect diversity and abundance and biomass across the globe. First one, pesticide usage associated with intensive agriculture. Secondly, pests like varroa mite, which you've probably heard about, that affects bees, wild bees, and also pathogens, also changing climate. But the really big one is habitat loss, through increasing urbanisation and clearing of land for agriculture.
What are the implications of that bigger loss within, I guess, our ecosystems?
Well, the implications are absolutely huge. If we just look at some of the services that insects provide, pollination, 85% of wildflowering plants are insect pollinated and 75% of agricultural crops. Insects also perform pest control services. Dragonflies eat mosquitoes, for example. Decomposition. Without beetles, we wouldn't have nutrient cycling. And food. It's not just tourists in Khe Sanh Road that eat insects. It's all the animals, the birds and the mammals that you see out there that also rely on insects as a food source. So yes, we should be super worried about the loss of insect diversity and abundance. It's kind of like a Jenga. Everyone knows Jenga, that game where you stack up the blocks. We can keep pulling out the blocks, but at some point we're going to pull out a block and there'll be catastrophic ecosystem collapse. And that's potentially the future if we keep losing our insects.
Well, there's the problem, Paul. But Josh, we're looking for solutions, aren't we?
There we are.
Tell us a little bit about your research. eDNA metabarcoding apparently is a Star Trek level technology that can detect insect pollinators and pests. Now, what is eDNA metabarcoding for those of us who don't know?
Sure. So I think the thing is that obviously when we talk about Star Trek level, we think of Spock's tricorder. And Spock with his tricorder could obviously detect anything that was in the environment. And eDNA is not too dissimilar. Environmental DNA really is these trace amounts of DNA that are left in soil, in water, air, and even on plant material. And using eDNA metabarcoding, we can actually amplify up these small little fragments, almost like forensics, and we can actually work out what organisms deposited that DNA. Now, what's nice is once upon a time, if you wanted to work out what had been in a certain area, you had to wait and watch it. You had to stand out there like David Attenborough and observe. But these days with environmental DNA, we can actually visit after the organism's been there and then see what's been there.
So I guess as part of research and understanding an environment, you need to know what's there. And obviously this is just an exemplary tool for achieving that, yeah?
Completely. And I think the thing is that, obviously when you're doing observations and monitoring, sometimes you want to detect one or two organisms, but because we can detect everything that's been in a certain area, we can actually detect entire communities, which is so much more powerful than just waiting there with one camera.
In terms of actually measuring loss, is that a longitudinal study that you're part of in terms of figuring out how an insect community is actually depleting?
Well, it's not part of the research that we did during my PhD, but it's certainly a longer-term goal of the work that we do in the Trend Lab is to actually incorporate more regular environmental DNA monitoring into a variety of systems. Agriculture is a really important one because it's not often looked at for biodiversity. When we think of agriculture, we think of honeybees, and we think of monocultures, and we think of systems that don't really have biodiversity. But insects are intrinsic to these systems. They are part of pollination. They're part of predators. They're also pests that cause huge crop losses. And so by incorporating eDNA more regularly into agricultural monitoring, that's where we're actually interested in doing these longitudinal studies that help us monitor losses over time to say, are our current agricultural practices actually sustainable?
Well, you've managed to, I guess, study a specific species within agriculture just to test your theory. Tell us about avocados.
Avocados, well, obviously great on toast, but also, when we were interested in doing this study, we were looking for a crop that was a really big producer in the state that relied on pollination, and avocados were a significant crop. And so we went down to Manjimup and Pemberton, and we were interested in actually using this technology to see if we could detect native pollinators from avocado flowers, and then also work out what native plants these pollinators relied on to see if we could show the importance of remnant vegetation to sustainable agriculture. And what was interesting is this research evolved as we went. We went there with one question, can we detect native pollinators? But as we went there and started using this powerful technology, we worked out that we could actually detect a variety of things, not just pollinators, but pests and predators that also interacted with these flowers. And so suddenly the question became much bigger, which is what's the entire community that's sustaining avocado orchards down in the Southwest?
I guess what conclusions are you drawing about these? I guess all of this this interrelated ecology that works together to for various results, such as an avocado crop.
Totally. So I think the thing is that what was most interesting when we started this research is that we went in there kind of like, can we detect honeybees? Are honeybees even interested in avocados? But then we detected that hoverflies are actually a huge flower visitor for avocado trees in the Southwest. And other research across Australia has also shown that hoverflies do visit avocado flowers. But then on top of this, we found wasps and calliphorids, which are blowflies. And so suddenly we were forming this much more complex picture of actually flies having a much bigger role in avocado pollination than just, the regular honeybees. And the research shows that honeybees are actually not that interested in avocado pollen. They don't particularly like the pollen. They don't love the nectar. And as a result, they're somewhat reluctant pollinators. But, the fly species that are present in the Southwest were ferocious with nectar and pollen on the avocado flowers. And visiting, we detected over 100 visits per hour for some of these species, especially the hoverflies when they were there en masse. And it just goes to show that there's actually a really diverse and rich community of native insects that are actually doing this job. And so although agriculture has traditionally been built around bringing honeybees with us, the colonialization idea of, we'll bring our ancestral pollinators with us, we're actually best to listen to the landscape and actually utilize what's there, because clearly there's amazing insects that are doing the job too. And we need to actually enhance their presence rather than just, bringing these old practices into the future.
I guess it just reinforces the importance of conservation of habitat and remnant bushland. That must be such a foundational thing, a foundational message that you're trying to get out there, Josh.
Oh, completely. And I think the thing is that, some of our research, we extended this because we were interested in looking at, could we then detect what plant species these insects are actually interacting with? And we found that, some species would travel out into adjacent bushland and actually harvest the flowers there and nectar and the pollen, and then come back into the avocado orchards. And so it shows that far from, this fence mentality, like an orchard finishes at the fence line and after that there's something else, this ecosystem is connected across the landscape scale. And when we're actually looking at an orchard, we have to think about what other things are present. And native bushland is often looked at as this uncultivated land. It's actually not utilized. It's got potential, but we could plant it out. So what should we do with it? But I think what, like you mentioned, the foundational message of this research is that the presence of native bushland is actually fundamental to sustainable agriculture, because it's those areas that actually make agriculture work. Unfortunately, in places like California, where they have massive avocado plantations, they have no remnant bushland anymore. And what happens is, although they've got ideal conditions for pollination, avocado trees are flowering, there's no pollinators. And so you've got to bring honeybees in, and there's this huge investment. But, from the research that we've done, we actually go that these areas of remnant bushland are just so diverse, and they hold so much of the insect abundance present in a landscape. If we don't harness those and we don't protect them, we're actually doing ourselves a disservice.
one of the ironies of the research that you've presented, Josh, is the very fact that the European honeybees that were brought over to do a job actually prefer, as you say, the native bushland. They're the ones that travel the most.
Yes.
To get away from
Totally,
orchards almost, right?
totally. And I think that's the thing is you've got to think about honeybees. They're efficient. They know their job. They are absolutely, amazing at sniffing out the best resources and then visiting them preferentially to avocado trees. And often when I talk to, farmers down in the Southwest, they would say, oh, the second eucalypt is over there flowering. The honeybees will just leave the orchard. I've got 10,000 avocado trees surrounding this one hive and they completely go past them because they're interested in, native eucalypts nearby. And it just goes to show that, honeybees, they will travel big distances and they can avoid, resources that they don't want to have to pollinate, which would be avocados in this case. And so, by utilizing other insects that are actually interested in avocados, like, the fly species that we mentioned earlier, there's this untapped potential that, we haven't really looked at in that way. But, by utilizing flies in this way, and some work by DPIRD with David Cook has also found similar things. They can be vastly improved pollinators for avocado trees, but also they stay local. They're not quite interested in traveling as far as honeybees, and maybe that's because they're not as efficient in the longer run. But, it just goes to show that there is a potential to use other species for a job that we've only kind of had a one-track mind of thus far.
But Josh, it's not just about pollination for the avocados. These native insects are also serving, I guess, a pest control function, right?
Completely. And I think the thing is that, we also detected, wasp species as well as, other native insects that actually moderate the presence of these, herbivorous insects that actually decimate crop productivity. And what's really interesting is that, farmers at the moment are left to rely upon their own personal observations. They'll walk up to a tree and inspect it for a species like six-spotted mite to see if it's present. And if they do detect it, then they'll undertake as a pesticide spraying for the entire orchard. Now that would be like one member of your family being sick and yet giving the whole family antibiotics. It's overkill. We don't need to do it to that level. And environmental DNA of trees within an orchard actually allows us to get targeted pesticide spraying because we can work out which trees have this pathogen or this pest on it, and we can actually target those particular tree species for these applications rather than spraying an entire orchard and harming predator diversity, which would actually benefit the orchard in the long run.
And I'd add that we can not only pick up the insects, we can also detect fungal pathogens, for example, that are vectored by the insects. So that's really, really important. So we can use the insects as our fungal pathogen sampler and incorporate it into an early warning system.
It's all this, I guess, understanding that you already have of these interrelationships, right? That come into play in this instance.
Yeah, absolutely. And I think some of Josh's work really emphasizes the importance of native vegetation as a pump for these insect species that perform pollination or positive predatory roles on other insects. So, farmers, need to consider maintaining native vegetation at their orchards or supplementing it by planting wildflowers amongst the rows of the orchard just to provide that habitat for native insects and maintain diversity of native insects at their location, which then provides redundancy in the system if some of the introduced pollinators like honeybees disappear because of something like varroa mite.
Paul, Josh's research has shown us a very pointed and very focused area in which eDNA has really opened up some new insights into pollinators. But what more broadly can eDNA tell us?
Yeah, there's absolutely huge potential for eDNA. All organisms contain DNA, so we can detect any species right across the tree of life. So we can look at everything from soil microbial communities to vertebrate species. And we can take a very targeted approach where we can look at specific species for a conservation outcome where we, say, want to detect a cryptic or rare arboreal species such as red-tailed phascogale in the Jarrah forest. So we can use DNA to complement traditional methods such as cameras to find that species, or we can look at whole communities of organisms, which is a big focus of what we do. So we'll look at change or delta in community composition. So we can look at, for example, the response of insect species to restoration. So if we restore a mine site, do those insects come back? Okay, so we need them to come back to perform all the ecosystem services that they, provide. So before that restoration, using a site is effectively restored, we need to be able to assess biodiversity at that site, and we can do that with eDNA. We're also looking at freshwater systems and marine systems. You can imagine, for marine, for example, that if you're working in the Kimberley, you want to do fish surveys to look at the impact of a pipeline in the sea. You don't want to get in the water in the Kimberley, or you'll end up being the meal of a crocodile. But we can use something like eDNA, take a liter of water, sequence that, and we'll be able to tell you all the fish species that are in that area. So incredibly powerful. It's a transformative and a disruptive approach that I think is just growing and growing in applications, and particularly natural capital accounting and the nature positive market. I think eDNA will have a big role to play in that, along with remote sensing, providing data for corporates.
Hypothetically, Paul, does eDNA enable, a provable quantitative measure by which you can almost set KPI, say, for a restoration project? I mean, can you, before a mine site exploration even takes place, can you, as part of the environmental assessment of the area, go in and use your Star Trek eDNA tools to determine what's there, and then do a comparison 10, 20 years down the track during the restoration as that progresses?
Yeah, that's exactly what I think the future holds. A company could invest in generating that baseline data on what's there as part of their environmental approval process. And then they can, revisit sampling in the same way, using the same methods as the project goes ahead. And then at closure of the project, to look at that recovery of biodiversity over time and prove to government regulators that, they have restored or not brought in pest species, for example, to a location.
You mentioned before that eDNA is a complementary method to existing techniques. In what ways is eDNA metabarcoding limited at all?
So like any monitoring method, it has limitations. So just as an example, one of the issues that we're having at the moment is we have a project in Bangkok where we're looking at urbanization impacts on bee diversity, native bee diversity in Bangkok. We're finding it very difficult to develop an assay, or assays like a molecular magnet, to pull out the DNA sequences from the bees that we're interested in, because bees are incredibly diverse. So it's very difficult to design an assay that's going to hit all bee species at a location. And also, there could be something unique about the ecology of bees, that they don't deposit much DNA when they interact with a flower. Some species may, some may not. So development of assays is a tricky one, is a tricky issue. So you can get false negatives for species. So species may be present, but you don't detect it through its DNA. So that's an issue. Abundance, a lot of ecologists, or a lot of environmental impact assessment needs abundance data.
Not just presence.
Yeah, not just presence or absence. So eDNA is not particularly good at providing abundance data, relative abundance perhaps in some situations, but that again is also a bit of a limitation. But as I said, all monitoring methods have got limitations. If you use cameras, for example, they don't pick up reptiles. They don't pick up small species. So every monitoring method has its issues. It's about understanding what the limitations of a method are, comparing it to traditional or long-standing approaches, so you can just calibrate your new method.
Josh, your study is done and dusted. Your thesis is in. You've got the letters. What is next for that paper? Is it being put in front of the agricultural sector? Is it finding a home somewhere?
Yeah, look, I think that's the most interesting thing, because, you develop a new method or you try a new approach, and then it's like, what do we have here? And I think at the moment, Steve Jobs has just walked onto the stage and he's held up this strange little black box and he said, I want to announce that this is an iPhone and this is going to change your life. And I think the thing is that we're at that stage. We've got this iPhone, but what do we do with it? And I think really the next thing for this paper and the next thing for this method in agriculture is actually to combine it with mechanization. You know, agriculture is really good at making things vastly efficient with robotics and technology. But we now need to combine this mechanization with environmental DNA metabarcoding, collecting measures of biodiversity simply as orchardists and farmers go about their everyday business. You know, having a tractor with an eDNA sampler on it, you know, collecting samples with some kind of, collecting device that is often used in agriculture as it presently stands. The thing is that, you know, when I did this study, I went out there with my family. You know, my granddad, he was out there helping me sample. And
Your granddad.
Granddad, hero. And, we were collecting all these flowers manually. Now, no farmer's going to do that, you know, when they're so busy. And so really the next step for this paper and the next step for this research is how do we combine it with currently used tools in agriculture to make it more accessible to the masses? When we do that, biodiversity will become a regular measure.
And can I just add in there, we had an honours project last year where we have developed a system to attach a DNA sampler to the side of a car. So any moving vehicle we could put a DNA sampler onto. So if you're driving through, you know, a farming area, you could put these DNA samples on a car or a header or something, and you could pick up potentially, you know, pathogens that are new to that area or insects or whatever that are invasive insect species. So I think that is just on the horizon at the moment. And also there's a potential to get real-time data with sequencing in the field.
There we go, Josh. Problem solved. I'm glad we brought the two of you together today.
Perfect. We did it.
Thank you.
What's next for you, Josh? What's what research are you undertaking now? Is it an extension of what you've been working on today?
Yeah, look, it's a really interesting point because actually, you know, I was fortunate enough to get a fellowship to Cambridge University. And I'm really excited to be actually heading north, and I'll be doing sampling in not only Cambridge, but also South Africa. And I get the chance to look for native pollinators in a desert ecosystem. So Curtin University has given me this wonderful platform to actually get on the world stage and try something completely new.
Josh, you must be absolutely thrilled. That's such a massive milestone so early in your career.
Oh, it was a bit of a breathtaking moment. You know, it happens to other people. And I think the other thing is that it felt like Willy Wonka's golden ticket. You know, like when I got that phone call, it really made me stop. You know, maybe there are a few moments in life like, wow, like my God, and that one it didn't feel real, and still it's sinking in. You know, I leave in March, and so it's gonna be a big life change, but I can't wait.
Congratulations, Josh. That
Thank you.
is fantastic, and a great outcome for the department as well, Paul. You must be pretty stoked with this outcome.
No, it's very exciting for Josh. We're very, very pleased for him. I'm planning to go and visit him in December.
It's gonna be busy.
Yes.
Now, look, you're both at different stages of your career in terms of the timeline. I'd like to hear from both of you, starting with you, Josh, in terms of the motivation for you to enter this line of work and to launch this career. What was this always something that was close to your heart as you were going through, I guess, high school?
Yeah, look, it's so interesting because in high school, you know, when they say pick your career, it's like, what does that mean? You know, what will I like? I come from a family of accountants, so you know, I wouldn't say passion is the main driver of many of our decisions. But for me, I looked at what I was good at in high school, and I was good at the one thing that no one liked, which was botany. And then from there, I remember volunteering at the herbarium, and the head of the herbarium at the time, I said to him, I want to do botany, but what goes with it? And he says, do genetics. And I was like, oh, genetics is gross. But I was like, if the head of the herbarium is saying it, he probably knows a thing or two. So that was university, and I did botany and genetics as my undergraduate degree. And then I looked at native pollinators when I was at Kings Park, and I was like, that's really interesting. And then COVID hit. And then after that, it was like, oh, you know, what do you do? And then Paul offered me this amazing opportunity to come to Curtin. And from there, it's how I, you know, evolved my passion for environmental DNA metabarcoding and looking at biodiversity more broadly.
You talk about an interest in botany that came about as a young fella, but was it also underlying all of that a mission, a desire to change, implement change, or something a little bit more lofty?
Of course. Look, I think, you know, for me, whenever I've talked to my family about it, a forest for me is the equivalent of a cathedral. there's something about when you're in these places, they're holy in a way. And I think that they're holy because they are representative of something that's untouched, and they are an area where it is just beautiful and they're so precious. And I think the thing is that we live in a society where it's so much easier to get caught up in an online momentum, trends, Twitter feeds, and then Facebook. And I think the thing is it takes us away from these special, special places where, these are what our biodiversity and what our world depends on. And we can't get distracted from that.
Paul, how do you see your role these days? From your early days in the field to now actually fostering new talent to go out there and continue the change?
Yeah, I really enjoy it. I think it's one of the most pleasurable parts of my job. I haven't actually been in this career for very long. I was an air traffic controller for a number of years and did economics and arts degrees way back and didn't even do science in high school, and somehow became a geneticist. But yeah, it's great to see people like Josh and many other students that we interact with going on and following their dreams and doing good things for the globe.
Well, Paul, you just, whipped out some, I guess, amazing career facts on your own behalf there. Was it a case of trying a few different things until you found genetics?
When I was 17, I didn't study science in high school at all. I wanted to do forestry but couldn't because I didn't study science. And then, just tried a few things, became an air traffic controller at a pretty young age. I think I was 20 or 21.
Wow. That's pressure. That's a real pressure gig for someone so young.
It's kind of like a sausage factory. The sausages are the aeroplanes and they come in and then they go out the other end and get packaged. But yeah, I got sick of that very, very quickly and decided to go back and do a forestry degree when I was early 30s and had 3 children, and then did a PhD, and by the end of that had 4 children. And yeah, I've never regretted it at all.
Yeah, I guess the potential for, an acting change, is that a really important part of why you do what you do?
Absolutely. I think, most of the people in our job are not motivated by salaries or, other things. But yeah, it's definitely that you can have a positive impact on biodiversity, and in particular Perth's biodiversity. We live in an incredible location with a biodiversity hotspot in Australia. Yeah, it's a special place and we have to do as much as we can to protect it.
Well, you're both doing such great work, and Josh, going from one biodiversity hotspot here in Perth to South Africa, which is widely considered another, you must be just glowing right now, mate.
Thank you so much, David. Yeah.
Thanks for your time today, both guys. Really appreciate you talking to us today.
It's been a pleasure.
David. Cheers.
You've been listening to The Future Of, a podcast powered by Curtin University. As always, if you've enjoyed this episode, please share it. And don't forget to subscribe to The Future Of on your favourite podcast app. Bye for now.